It is my distinct pleasure to get out of the rain and introduce Florence Varodayan, who is a postdoctoral research associate at Scripps Research Institute and a faculty candidate here at the School of Neuroscience. So Florence has been working on the study of alcohol and alcoholism throughout her scientific career. She started working with her PhD with Neil Harrison at Columbia University, where they looked at some of the cellular aspects and molecular aspects of alcoholism using cell culture models. After getting her PhD, she went to the Scripps Research Institute working for Marissa Roberto, where she actually studied using electrophysiology and behavioral techniques to start looking at circuits and more behavioral aspects of alcoholism. In the course of this research, she has won 5 national and international travel awards. She has chaired 2 symposium on alcohol. She has 12 invited talks to talk about her alcohol work. She has 23 publications on alcohol. If there's any questions that you have about alcohol addiction, she is probably the most knowledgeable person in the entire city of Blacksburg right now. And I would say she also gives a fantastic talk. And it's my pleasure to introduce my colleague and friend Florence for her wonderful talk. So please give a warm introduction.
Thanks, Matt. So what Matt failed to mention is that we overlapped for a couple of years in the same lab at Scripps. And as he and I just discovered, despite all those publications and despite all those publications, we managed to be in the same lab for 3 years and not publish together. So I think we need to collaborate. But thank you all for having me here today. As Matt said, my name is Florence Varodayan, and I'm a postdoc at the Scripps Research Institute. And my interests lie at the intersection of alcohol and stress. And the brain has several major stress signaling systems, and it's become very clear that alcohol is able to dysregulate some of these systems, and this leads to the progression and the development of alcoholism, as well as the maintenance of these alcohol use disorders. So, as everyone in this room is well aware, alcohol is an anxiolytic. People consume alcohol as a self-medication to cope with stress. In addition, stress is one of the primary triggers for why a person who's an alcohol-dependent individual is unable to maintain abstinence. So they start their abstinence period, and then they experience stress-induced alcohol cravings, and that leads to subsequent relapse. So there's a great clinical need for identifying compounds and identifying novel targets that we can then use compounds to treat to allow alcoholic individuals to maintain their abstinence. These are people who want to be abstinent but struggle to maintain it. And this has been the focus of my postdoc and my graduate work as well, actually, trying to mechanistically understand how alcohol is dysregulating these brain stress systems with the idea that then we can start to think about druggable targets. So what is an alcohol use disorder? Many people socially drink in the US and globally. But only a small percentage of these people go on to develop, well, I shouldn't say a small percentage, a fair number, but not everyone who socially drinks goes on to develop alcohol abuse issues or alcohol dependence. And more importantly, a person who becomes alcohol dependent doesn't become alcohol dependent after one drink. It's a gradual progression. People begin to drink more often and more heavily as they transition to this alcohol dependent stage. So alcohol abuse and dependence can be thought of as a continuum, with these different stages progressing into each other. By the time someone becomes alcohol dependent, they've had these neurobiological changes in their brain as a result of this chronic exposure. And as a result, they when they try to become abstinent, they experience withdrawal. So they have physical symptoms of withdrawal, they can have seizures or tremors, but they also have an emotional cognitive effect of withdrawal, where they might experience hyperalgesia or alcohol cravings or stress and anxiety as a result of the withdrawal. So this is why I'm really interested in understanding the intersection to understand how we can treat it. So when you think about that, the important thing to think about is, why do people drink? And as you progress from being someone who's alcohol naive to someone who's a social drinker to someone who's an alcohol-dependent individual, the motivation to drink changes. So when you're a social drinker, when you drink alcohol, it stimulates the reward circuit in your brain, releasing dopamine, and it's a pleasurable feeling. As someone progresses into the alcohol dependent stage, this changes. And now, instead of feeling the pleasurable effects of alcohol, what they're actually feeling is that when they don't drink, they have very negative emotions. And so they're now drinking to counteract that negative affect and those stressful cravings and that stressful withdrawal symptoms. And so they're essentially drinking to ward off the negative consequences of being abstinent. And this suggests there's a change in the neurobiology of the person and a change in perhaps the circuits that are being recruited by alcohol in someone who's a social drinker versus someone who's an alcohol-dependent individual. So when you think about the progression of this disease, and you really try to think what's happening on a neurobiological level, there's lots of different factors that can go into someone developing alcohol dependence. There's lots of risk factors. So there's things like genetics, epigenetics, early life stress, life experience. But critically, during this transition stage, they go from a situation where alcohol feels good to alcohol being something they need to drink to feel normal. And associated with this has been shown to be an increase in stress axis dysfunction. And this stress axis dysfunction is hypothesized to be the basis for this anxiety and stress and craving in an alcohol-dependent individual. So this leads to my central question of how do alcohol and stress interact to promote alcohol use disorders? What are the neurobiological changes that are happening in the brain of a person who's alcohol dependent that causes them to have these negative emotions? And these changes are actually quite long lasting. So alcohol-dependent individuals have been abstinent for years and years and years, still report what should be neutral stimuli as being stressful. They still report stress disturbances 8-10 years later. So these are very long-lasting changes, and they make it very difficult for them to maintain their abstinence. So not surprisingly, the brain circuitry that's involved in the different stages of the disease overlaps significantly with some of the brain regions involved in stress. So in red here, sorry, my pointer, in red here, you can see the extended amygdala circuitry. And this circuitry is actually involved in the immediate abstinence period when an alcohol-dependent individual is going through withdrawal and enduring stress-induced cravings and anxiety-like behavior. In contrast, this green circuitry highlighted here is cortical circuitry and hippocampal circuitry. And that's the circuitry that's thought to be involved in alcohol craving. And a particularly insidious part of this disease, which I've kind of alluded to already, is that someone who's alcohol dependent or addicted to other drugs of abuse actually has this really strange phenomenon where the longer they maintain abstinence, the harder it is for them to maintain abstinence. And it's because these different brain regions, when you do brain imaging studies, actually light up more in these brain imaging studies and show greater activation the longer they've been abstinent when they're put in stressful situations or shown drug-induced cues. And I think this is really the crux of the matter when it comes to treating alcohol dependence, is that the longer you're successful at staying away from the drug, the harder it actually is for you to maintain it. And that's where I think we want to try to find treatments, because a lot of people do want to recover from the disease and they want to maintain their abstinence, but they really struggle with it. So today I'll be talking to you about two of my research projects that I've been doing as a postdoc and then as I've beginning to transition into my independence. And these projects are focused on alcohol's dysregulation of stress signaling systems in the brain, and particularly in the central amygdala and the medial prefrontal cortex, and how these different signaling systems and their dysregulation lead to deficits in motivation and cognitive deficits. And I realize going back, I should point out those regions to you. So the central amygdala is located here as part of the amygdala complex, as part of the extended amygdala complex. And the medial prefrontal cortex is part of this circuitry that's involved in the preoccupation and the anticipation of alcohol when you're craving. So starting off with, how does the amygdala CRF signaling drive the development of alcohol dependence? And this is work I've done primarily as a postdoc in the laboratory of Marissa Roberto. And Marissa Roberto's laboratory is very interested in the central amygdala. It's a really interesting structure. It's a primarily GABAergic nucleus. So it has GABAergic interneurons, and it has a GABAergic projection neurons. So the projections it sends out are actually inhibitory and inhibit downstream regions that are involved in addiction, such as the ventral tegmental area and the lateral hypothalamus. This region is thought to be a hub for alcohol and anxiety and stress, as it's activated by stressful events, these from brain imaging studies. And interestingly, it is a region that is very important in alcohol use. So alcohol-dependent individuals have an overactivation of their central amygdala. They show greater volume of amygdala structures, and they show a greater amygdala-based fear response to what should be non-threatening stimuli. And as I mentioned earlier with the insidiousness of this disease, the longer they've been abstinent, so 1 week versus 1 month versus 1 year, the greater that overactivation and the more it lights up in these brain imaging studies when they're showing something that should be a non-fearful stimuli but is fearful to them. In addition, the central amygdala, to do with this overactivation, actually has various stress peptides that are available in the central amygdala to regulate its activity. And it also has, it's a bit kind of low on the screen, but several anti-stress peptides. So the amygdala is this really interesting hub where you can have pro-stress influence and anti-stress influence, and really how that calculates out and the summative effects of that is what leads to the overactivation of the structure and the downstream effects to promote alcohol dependence. And so the region's a really unique region because it has all these different opportunities to modulate activity. And the idea is that if we can modulate the activity using one of these neuromodulatory systems, we can then start to think about treatment. So can we dampen the effects of some of these pro-stress peptides and neurotransmitters, or can we enhance the effects of some of these anti-stress molecules? And in particular, corticotrophin releasing factor has been of great interest. So systemic administration of corticotrophin releasing factor, or CRF, as I'll continue to refer to it from now on, has been shown to reduce drinking in alcohol-dependent rodents, and it's been shown to reduce anxiety-like behavior, both in alcohol-dependent rodents and in rodents that have been exposed to other kinds of chronic stress. So that's a really promising peptide to think about when you think about how we can regulate central amygdala activity to dampen this overactivation and perhaps slow the transition to dependence or alleviate some of the long-term effects of that overactivation in people who are trying to become abstinent and stay abstinent. So all of this background, when I entered the laboratory in Marissa's lab, led me to the hypothesis that alcohol heightens the influence of the CRF signaling system in the central amygdala, and this promotes the withdrawal-associated behaviors and pathophysiology of the disease. So that led me to think about 3 specific questions I wanted to answer during my time in the laboratory. How do alcohol and CRF interact to regulate CEA activity? Do the mechanisms of how they interact impact alcohol intake and other withdrawal-associated behaviors? And given the first 2, in the long term, is the CRF system dysregulated, and how is it dysregulated? Because only by knowing how it's dysregulated can we think about how to translationally change what's going on. So to do these studies, we use a model of alcohol rat dependence. And in this model, it's a standard vapor chamber model. It's used in several labs around the country. I'm not sure if Matt has vapor animals set up. It looks like he does, based on the nodding. And so in this model, we take adult male Sprague-Dawley rats, and we expose them to 5 to 7 weeks of alcohol vapor. So each week, every day, they get exposed to 14 hours of ethanol, and then they have 10 hours off. This happens every day for 5 to 7 weeks. And we're looking to achieve a target blood alcohol level of 100 to 200 milligrams per deciliter, which is equivalent to 0.15%. So 0.08% is the drink driving limit, to give you an idea of how that translates. So this is a relatively high level, but it's definitely a level that is achieved by people who are drinking heavily and drinking often. And previous work from our laboratory and other labs has shown that animals that go through this model for 5 to 7 weeks show physical symptoms of withdrawal that mimic the human condition. So they show tremors, they can show seizures in extreme cases. They also seem to show some of the same emotional, psychological effects. I mean, as much as you can have psychological effects in an animal, but they seem to show some of this, mimic some of the same things that the humans have. So they show hyperalgesia, they show withdrawal-associated stress, they show elevated brain reward thresholds. And so it really seems to be a good model for generating animals that can then have predictive value for the human condition. And this is just an example of the setup. So we have these vapor chambers. The animals go into the cages, gets closed up, and they get exposed to vaporized alcohol for the period of exposure. So on the last day of exposure, we take these rats, and I'm primarily an electrophysiologist. So what I'm doing is I'm sacrificing the animal, I'm slicing up the brain tissue, and I'm interested in looking at what's happening in terms of neural communication, so cell-to-cell communication across synapses, and specifically looking in the central amygdala. So just to give you a schematic of what I do for all the students and non-electrophysiologists in the room, what I'm actually doing is I'm taking a glass pipette and I'm literally poking a cell, an individual cell, visualizing under a microscope. And what I'm able to do by doing this is listen in to the cell's activity. So the cell has electrical conductances, and that's how the cells communicate, and then they have the chemical conductance over the synapse. And so what I'm able to do is listen in to how the cells are communicating with each other and basically understand if that's different in a naive animal versus a dependent animal. And this is an example of what my output looks like. So each of these little ticks is actually a current. So it's actually a representation of neurotransmitter crossing the synapse and opening ligated ion channels to allow chloride in, because this is an inhibitory situation. So I'm looking at GABA transmission and GABA currents. The frequency of these little ticks down is actually equivalent to GABA release. So a higher rate of ticks means that there's more GABA being released, while the amplitude and the kinetics of those are related to GABA A receptor function. And in this talk, I'll be focusing exclusively, in this section at least, on GABA release, because that's where I've seen the most changes. So going back to my question, how do alcohol and CRF interact at GABA synapses in the central amygdala? And so first I bath applied ethanol, and I found that ethanol increased the frequency of SIPSCs, so it increased GABA release directly on the slice. And CRF did the same thing, independently bath applied. When I blocked CRF using a CRF1 antagonist, ethanol was still able to increase GABA release, suggesting that the two systems don't interact in the naive rat. However, in the dependent animal, when I applied ethanol, once again, same thing, increased GABA release, and same thing for CRF, increased GABA release. However, when I used the CRF1 antagonist, ethanol's effects were now dampened. And this shows that now alcohol is actually working through the CRF1 receptor to have its effects on GABA release, as depicted here. So in the dependent situation, alcohol seems to be working through this receptor. To better understand this switch-in mechanism, I then did some studies pharmacology studies, looking at other intracellular factors that could be involved. And I found that, so this is the ethanol effect in naive animals. In naive animals, alcohol works through L-type calcium channels and works through the IP3 receptor to increase its GABA release. Whereas in dependent animals, alcohol works through the CF1 receptor, as I already showed you. And now you can see that the ethanol effect is blocked in the presence of the pKa inhibitor and the PKC inhibitor, showing that it works through that pathway, as well as the ryazine receptor. So essentially, I've identified two different mechanisms. In naive animals, alcohol is having its effects on GABA release through an L-type calcium channel mechanism, while in dependent animal, it's now working through a CRF1 receptor-based mechanism. And this is really interesting because even though alcohol is having the same outcome of increasing GABA, it's now doing it through different mechanisms. And it seems to suggest that in the dependent animals, there's a sensitization of the GABA synapses to the CRF1 signaling system. So this led to my next question of, do these different mechanisms have an impact on drinking? Because after all, I'm interested in translational work. Is there an effect on the pathophysiology of the disease? And to do this work, I collaborated with Dr. Giordano Di Guglielmo, who was a postdoc in Olivier George's laboratory at TSRI. So in this study, we wanted to look at how alcohol is affecting animal behavior. And so the model we used was a self-administration model. So in this model, the rat is trained to self-administer alcohol. And after it's been trained to self-administer alcohol, once all the rats have been trained, we then put them into the vapor paradigm. And in this way, we can look at differences in how the animals self-administer after they've acquired alcohol dependence, and if we can change that by using these different antagonists. So first off, this is a video of an animal self-administering alcohol. And it plays, there we go. So the animal is pressing the lever down here, and then when the light turns on, it knows it's getting its reward. It goes to the sipper tube, sips some alcohol. After a timeout period, it once again can press the lever. The light goes on, and it goes over and samples its alcohol again. And what's really nice about this intake system is that after vapor exposure, the alcohol-dependent animals actually self-administer significantly more alcohol. And so now we can look at what is happening in the brains at the circuit level of animals that have low levels of exposure, which can mimic our social drinkers, or animals that have this vapor exposure, which then causes them to seek out higher levels of alcohol. And so the next thing Giordano did was infuse these two different antagonists. So an antagonist for the L-type calcium channel and an antagonist for the CRF1 receptor. And he fused each of them into the central amygdala and looked at how it affected the drinking and the lever pressing of these individuals. And so first of all, in the non-dependent rats, so the rats that haven't had vapor exposure and are mimicking our social drinkers, Giordano found that when he injected in nifedipine, the L-type calcium channel blocker, there was a reduction in the number of lever presses the animal did and the amount of reward it received, while there was no effect of the CRF1 receptor antagonist. In contrast, in the dependent animals, first of all, here's their baseline lever pressing. This is their lever pressing once they've been made alcohol dependent. And with the L-type calcium channel, there's no effect on lever pressing. With the CRF1 receptor antagonist, now we see a reduction in lever pressing. So what we've essentially shown here is the electrophysiological studies that identified the mechanisms. The L-type calcium channel mechanism in the equivalent of the social drinking or naive animals, and the CRF1 dependent mechanism in the equivalent of in the dependent animals. These two mechanisms translate to the behavior. So if we block the behavior based on the mechanistic if we block the receptors based on the mechanistic studies, we see changes in behavior. So we see a reduction in this escalation of drinking in the dependent animals. And this is illustrated here. So this L-type calcium channel mechanism in naive rats governs alcohol's effects on GABA release and governs the drinking behavior of the social drinking equivalent animals, while the CRF1 receptor mechanism in the dependent animals governs the escalated drinking of the dependent animals. And this was really exciting because, first of all, it's the first time the L-type calcium channel was implicated in social levels of drinking, but it also really suggests that we're on the right track when we're thinking about CRF dysregulation in the central amygdala as a driving force in the development of alcohol dependence. So this actually led to the third question, which is, okay, we know that alcohol is dysregulating the CRF response by hijacking it, essentially, in the alcohol-dependent rat. But how is this dysregulation happening? How is this chronic alcohol exposure affecting CRF signaling? And so we started to think about the levels of CRF in the central amygdala. And CRF is a really interesting stress peptide because it's primarily released by the hypothalamus as part of the HPA stress axis. But there are also lots of different centers of extra hypothalamic CRF. In particular, the central amygdala itself has a lot of local CRF release, and there are also CRF inputs from multiple other regions, some of which are depicted here, such as the BNST or the insula or the medial prefrontal cortex. For this part of the study, I collaborated with Dr. Candice Conte, who is a professor at Scripps. And she actually had some preliminary data in her mouse model showing that local CRF CRF is released locally in the central amygdala, but that source of CRF is not the source of CRF that governs drinking. So we started to think, okay, if it's not the local CRF, it could be one of these circuits. And the circuit we were most interested in is the circuit that goes from the parasubthalamic nucleus to the central amygdala. And this is an extremely neglected circuit. There's only 3 or 4 publications on PubMed about it. No one had recorded in this region, so it was very exciting for me as an electrophysiologist to think about going to this region and seeing what we could do. And so what we did in these studies is, first of all, Candice wanted to confirm that in our hands, we do see a CRF-positive neurons in the parasubthalamic nucleus projecting into the central amygdala. So she injected into her CRH iris Cree mice a virus, and this virus, which is written up here, essentially what it does is it expresses several fluorescent proteins. And so at our injection site in the PSTN, only in CRF-positive cells do you get expression of TD tomato. So we can identify each of these little cells we know is a CRF-positive cell in this subregion. In addition, because it has this fusion protein of synaptophysin, which is a presynaptic terminal protein, fused to a GFP protein, might be a little bit hard to see, but all of these little puncta, this image is taken from the central amygdala, and all of these puncta represent the fusion protein in the central amygdala. So this tells us that there are CRF projecting neurons in the PSTN that project to the central amygdala, confirming that this, in our hands, in our mouse model, we do see this projection. So the next thing we wanted to do was to look at the activity of this projection. And we started off doing that by using inhibitory dreads. And so what we did is we injected this inhibitory dread virus into the PSTN, and we wanted to look at how this circuit affects drinking behavior. And again, this was in the same mouse model, the CRH iris Cree mouse. So these channel rhodopsin are only expressed in the CRF-positive neurons in the PSTN. And using the mouse model, we once again saw that in naive animals versus animals that had gone through the vapor chamber model, we see an escalation of their alcohol intake. This was actually 2-bottle trace this time. And we found that when we systemically injected CNO at 2 different concentrations, there was a reduction in the alcohol intake of the dependent animals across both doses and in the naive animals across the higher dose, the 3 milligrams per kilogram. So this indicated to us that these PSTN CRF positive neurons are able to regulate drinking at the lower dose in the dependent animals and at the higher dose in both groups. So then to ask more about how this circuit is functioning, I then took these animals and I did my slice electrophysiology of them. So I started recording from M-cherry positive cells in the PSTN. So these are the cells that we know are projecting to the CEA. Or, sorry, we know they could be projecting to CEA. They also could project to other regions. And what I found, first of all, is confirming the effects of the CNO. When I applied it directly in the bath, I was able to reduce the firing rate of these M-cherry positive neurons. And really excitingly, when I compared the effects, when I compared the firing rates of these M-cherry positive neurons in naive animals versus dependent animals, there was actually an increase in the excitability of these cells in the dependent animals, indicating that our hypothesis is headed in the right direction. So in the dependent animals, the cells that could potentially project to the CEA are more excitable, suggesting they might release more CRF into the region. One of the big caveats of this study is, of course, these neurons don't just express CRF. They're glutamatergic neurons, so they obviously could be releasing glutamate in the CEA, but they also express several other neuropeptides and neurotransmitters such as substance P. And so the next stage of this study is that we're injecting the excitatory dread into the PSTN and looking at its effects behaviorally and electrophysiologically. And Candice has shown already that in naive animals, when you inject this excitatory dread and you give the animals CNO, you can actually escalate drinking in a naive animal. So the next stage will be now for me to record from the central amygdala and use the different pharmacological receptor blockers to identify what peptides could be involved in that process.
Question.
Sure.
So you were saying, I don't think you showed it, but I think you mentioned it, that you think that the CRF coming from the PSTN, but not that local CRF within the CA
Right.
is mediating that? Do you know, like, what?
Oh, yeah, sorry. So I was not involved in that part of the study, which is why I skipped over it. But no, it's a great question. So Candice had done so the original idea was that it would be the local CRF. I mean, I think that was the obvious idea, and it made a lot of sense. But Candice had done some preliminary work where she'd actually put the dreads into the CEA. And she found that when she blocked local CRF release in the CEA, she didn't see any effect on these drinking behaviors as well as anxiety-like behaviors for those studies. And so it was actually quite surprising that she didn't see that. And so this is what led to our literature search and our idea to go after this neglected circuit. Yeah, so it's an excellent question. Are there any other questions before I move on? Okay, great. So so what we're really interested in doing is trying to tease apart now that we think this circuit is involved, trying to confirm this circuit is involved and really understand, is it CRF? Or as I said, we had an unexpected finding that it wasn't local CRF. It could also be a different peptide, and the electrophysiology will help us come to a better understanding of that. So overall in this part of the project, what I've shown you is that I've shown you a mechanism by which alcohol recruits GABA release in the central amygdala in naive animals, an L-type calcium channel mechanism. And this mechanism can actually govern the drinking of non-dependent animals or animals that are equivalent to social drinkers. In dependent animals, we actually found that alcohol now recruits a different pathway that's actually a stress pathway. And so essentially what alcohol is doing is dysregulating the CRF stress system. And we're starting to form an idea of how it's dysregulated in terms of perhaps that input from the PSTN. And the impact of this dysregulation is this escalated alcohol drinking. And by antagonizing this system locally in the CEA, we can actually reduce that elevated drinking. So this work, in combination with the other work of other people that's shown that CRF in the CEA can also have important effects on anxiety-like behaviors, would suggest that it's a really good target for translational application. And that would be really exciting if it was true. But unfortunately, there have been several clinical studies done in the last few years that have failed to find an efficacy of the CRF1 antagonist on the alcohol craving and the stress-induced imagery effects in alcohol-dependent individuals. And that was really disappointing, actually. I think for the field as a whole, I think the field was converging, and the field really thought that we had found a new drug. There are very few drugs out there to help people who are alcohol dependent, and we really thought we'd hit on something. And so it was quite disappointing. But I think there is a silver lining, because I think that the realization is that it's not that the drug didn't do what we expected, but maybe we're not treating the right population right now. Go ahead.
Or is it because, I mean, CRF, those receptors are in lots of places in the brain, so
Oh,
maybe
yeah.
you needed to hit that specific cell to
Right, absolutely. So yeah, so this is exactly what my thinking is that there's multiple things. So it could be unique subpopulations. So maybe there are certain types of alcoholics that this would be effective in. And I'll answer your question absolutely in a second. But one example of that is one of the other drugs that's used to treat alcohol. One of the few drugs that's used to treat alcoholism is Naltrexone, which is actually also used to treat some opioid addiction, and it's an opioid receptor blocker. And when it went through clinical trials, it did very, very well. And what doctors found when they started prescribing it is that it wasn't working as well in certain populations. And it turns out that it's based on a genetic variant in the OPRM gene, which is the receptor gene. And as a result, it happens to work very well in Caucasian males, which was what the clinical trials were done on back in the day. And it's turned out that it doesn't work as well in females. It doesn't work as well in other ethnic populations. So it could be a population issue. But absolutely, the other side of it is that it could be that we need to hit this circuit in a more specific way. And that maybe, and that's where my interest in understanding mechanism really comes from. Because if you can understand mechanism, maybe you have the right idea, but you're not quite hitting the right druggable target. And if you can understand the mechanism, then you have an option of druggable targets. And I think that's where my interests really lie.
So what if the humans just respond differently than rats or mice?
Oh, absolutely. So yeah, the idea of, is this a good model? Does this have predictive value? That's an absolutely big concern as well. It's something I think the field has thought about a lot recently. We've had a lot of success with this model and predictive value for other things, but it's entirely possible.
I mean, for once, we're not inhaling alcohol. Most of us don't, right?
Yes. So the reason we use this model, go ahead.
If you just have them drink, usually rodents don't drink. I mean, it's hard to get them to drink.
That's not entirely true. So I will say two things. First of all, the field has come up with ways to get rodents to drink. I think the thing is that rodents don't like the taste of alcohol, which is the same as humans, right? Most humans, when they start to drink alcohol mixed with a lot of sugar. They're drinking cocktails, they're drinking Mike's Harbor lemonade, and then they progress to their fine scotches and, their fine rums.
As Germans, we drink beer.
Okay, fair enough. But so if you start with things like a sucrose alcohol mixture and you fade out the sucrose, you can train animals to drink. Absolutely. The value of this model is that there's two values. First of all, we can, by using the vapor model, we are actually controlling the amount of alcohol the brain is receiving. So we can regulate the blood alcohol levels of the animals. And so we can create a very homogeneous population. The second thing is that by giving these animals chronic alcohol exposure, the vapor model, they then choose to consume more themselves. So with the self-administration paradigm, the animals are actually choosing to press the lever and have higher intake. And they do the same thing with two-bottle choice.
Wouldn't one want to model sort of the cyclical elevation and decrease in elevation in ethanol levels that the typical alcoholic would have? I mean, even Isley, and so therefore their alcohol level in the blood will actually decrease versus these, when they're actually exposed to the vapor, it's constant, as you say, it's a flat line.
So actually, I'm glad you pointed this out because evidently I did not explain it properly. That is actually the value of this model. Because we have the 14 hours of exposure, they reach high blood alcohol levels and then they withdraw. And then they reach high blood alcohols and they withdraw. And that's actually thought to be really important to the development of the disease itself. So that's actually what we're modeling. You could also model that with volitional drinking. So once you train animals to drink alcohol, you can do paradigms where you teach them that they're only going to have a very short, predictable amount of time of access to alcohol, and they will binge drink. Whereas if you give the animals 24 hours, they'll have sort of a flat line, a flat low level. If you give them these very short amounts of time, they will binge and then withdraw and then binge and then withdraw. So that's something we are doing as a field. Sorry, go ahead.
So the self-administrating part, they're actually drinking the alcohol, is that right?
Yes. So what they're doing is they're pressing a lever, and then they, when they press the lever, they receive a reward, a drop of alcohol through the sipper tube, and they drink it, and they press again, and they drink it. So they're choosing to drink the alcohol.
So in that context, there must be a little bit of, you're talking, I was wondering about this, about the taste aversion for the ones that haven't been exposed to the vapor versus the vapor ones. There might be a taste aversion too, but they're some sort of drug effect. So there could be two things going on there, right? A combination of taste aversion and not getting the drug hit. That's my first. And the
Right.
second question I had was, which I also probably missed, is it CRF acting locally as well as PSTN, or do you think it's just PSTN acting alone? Do you still think, even though the local isn't sufficient, but do you think both? Do you have some idea if both are necessary to
Right. So to answer your first question about taste aversion, so before any of the animals get exposed to vapor, and this will be true of the next study I show you, they're all trained to self-administer alcohol. So you all have to reach a baseline of ability to self-administer alcohol before we expose any to vapor. So we are controlling for that. And then to address your second question of local versus external CRF.
Or if it's a combination.
Right, or a combination, exactly. So I think something that I think about a lot as an electrophysiologist is it is true that there are elevations in CRF throughout the central amygdala, but there's also most likely local hotspots. And I think it really matters where the CRF has been released within the network. So which synapses on which cells. And so that's something that's very hard to tease apart. But I think this is where like using techniques like DREADS or opto can really help us, because it gives us some more specificity in how we control the circuits. And those are the types of questions we're trying to ask. So right now I don't have the answer. I can tell you that local CRF alone is not enough to affect drinking or anxiety-like behavior. And I can tell you for sure that the PSTN projections, CRF positive projections, are enough, but how those intersect, I can't tell you yet, but that's definitely something we're thinking about. And also those, as I mentioned, those CRF positive projections from the PSTN also express a lot of other peptides. So, and I told you that the peptides are very important in CA, so like teasing that apart is definitely the next step. Go ahead.
Along the same lines, you're recording from the presynaptic GABAergic cell, but ethanol has been known to be an allosteric modulator of GABA-A receptors on postsynaptic cells.
Right.
So how does this really integrate at a circuit level? It looks like you have increased GABA release and you have a prolongation or a potentiation of the postsynaptic GABA response. Have you ever looked at the
So I'm actually, the measure I'm using, I'm recording these spontaneous inhibitory postsynaptic currents. We don't see changes in GABA-A receptor function in this region. Oh, that's not true. We see them in some cells, but not consistently in all cells. But it's certainly something to think about when you think about network integration. And so I think it's something we definitely think about because, and not just that, but alcohol is a very strange drug in that, as opposed to other drugs where, you know, there's a drug and then there's a receptor. Alcohol acts on a lot of things. You know, it acts on, I'm sorry, I don't know why my computer keeps doing that. It acts on NMDA receptors, it acts on GABA-A receptors, it acts on GABA-B receptors, it acts on so many different types of GPCRs. And so that's where I think the complexity of studying alcohol comes from, is it's having so many multidimensional effects at the same time. But that's why I think it's so important to think about what is the overall effect on the region or the circuit, and it also gives us the power to think about how to control it better. But it's absolutely something that we think about and we pay attention to because it's having very varied effects. Go ahead.
Given the interaction with GABA receptors and ethanol, have you seen any differences in the females across their estrous cycle with these rats?
So what's really interesting is we haven't done this type of work directly, but what we have started to do is look at just drinking across the different stages of cycle. And it does seem that females do have different effects in terms of how they drink, the levels they drink, depending on their stage of cycle. And that's something that's becoming more and more looked at and more important. And I also talked about unique subpopulations, and that's also where this is going to come into in terms of, there's also human data that, self-reported human data that suggests that women do alter the amount they drink and the patterns with which they drink across the different stages of their cycle. But this is also affected by social factors like a weekend versus a weekday and things like that. So it's something we're starting to think about more, but in this particular model, we haven't done this yet. But it's definitely something of interest. Any other questions before I move on? It's actually perfect time because I'm going to move on to my second project. So, yeah, go ahead.
40 millimolar ethanol, how much is translated to percentage?
Oh, that's a really great question. So 17 millimolar ethanol is equivalent to 0.08%, which for most humans is about 2 to 3 drinks. So 44 millimolar ethanol is like, let's say 6-ish drinks, which is high for a person who's not an alcohol-dependent individual, but is a relatively low level for an alcohol-dependent individuals. Some alcohol-dependent individuals will drink 24 beers in a day, you know. So it's not an unheard of concentration.
So you think the ethanol acts directly on the L-type and the CRF receptor, or
Mhm.
just some upstream effector signals to this?
Oh, that's a really great question. I honestly cannot say. That's a really interesting way to think about it. Yeah, I really don't know. That would be the only way to tease that apart would really to be to happen to stumble upon whatever that upstream mediator is and then do the co-pharmacology experiments. But I think it's a really important question because, as I mentioned, the more elements we know in these mechanisms, the more opportunities we have to find druggable targets that may have more direct effects or may not have off-target effects or may be more effective. Anything else? Okay. Oh, sorry, Matt. So as I was saying, we've identified these two different circuits, and I think this is a representation of the types of questions I like to ask, and sort of my approach to doing science, sort of thinking about how does mechanism affect specific synapses or circuits, and how does that translate into behavioral outcomes that we can then try to manipulate and rescue, essentially, or recover back to the normal phenotype. And so as I've progressed through my postdoc and started to think more about independence and what I want to do in my future laboratory, I've become very interested in a different region, the medial prefrontal cortex. And my interest in this region really stems from some of the human data, once again. So alcoholics show reduced prefrontal cortical volume. And they also show major deficits in cognitive function. So deficits in impulse control, risky decision-making, deficits in emotional processing. And all of these factors actually are really detrimental because it seems that the chronic alcohol exposure is leading to this impulse control issue. But then, of course, impulse control issues are something that is going to promote the progression of the disease. And, sort of it's a downward spiral from there. And so thinking about how we can alleviate these symptoms is really important. And there's currently no medications that are able to treat these types of symptoms in human alcoholic patients. So when I thought about cortical dysfunction, once again, inspired by this idea of translational work, I've looked at the literature on what is in the pipeline and what is of interest to the human laboratory studies in terms of modulating cortical function. And norepinephrine is a really interesting transmitter in this regard. So norepinephrine is a stress transmitter, and it's released at basal levels in the medial prefrontal cortex. And it's a really interesting neurotransmitter because it governs cortical function or dysfunction. So to have optimal function of your cortical processing, you need a certain amount of norepinephrine. And what actually happens is you have that basal level, and then when you encounter a stress, you actually have a stress-evoked increase in norepinephrine levels. And that actually leads to a decrease in cortical function. And so you can see that from this schematic that depending on the basal level that you have to begin with, you can actually have profound differences in the effects that stress or arousal can have on the system. And so what I've been thinking about is how is this system dysregulated with alcohol? So perhaps an alcohol-dependent individual, you already have an increased level of norepinephrine release in the brain. And so you're already at some kind of impaired function too far across the curve. And can we bring that back? And so this led to my hypothesis that alcohol is dysregulating the noradrenaline system in the medial prefrontal cortex. And this might be the cause of some of the cognitive impairment function issues, as well as the effects of the cortical projections to other regions like the central amygdala and those anxiety-like behaviors. So this led to my overarching question about what role does noradrenaline play in alcohol-induced cortical dysfunction. And more specifically, the two questions I asked in my funded work, so this work has been funded by K99R00, is how does alcohol alter a noradrenergic influence over this circuit, the medial prefrontal cortex, the central amygdala circuit, and does this dysregulation contribute to cortical dysfunction and anxiety-like behavior? So to do these experiments, I started by, once again, trying to trace the circuit. So I injected these retrograde tracer beads directly into the central amygdala, and I looked for M-cherry positive cells in the cortex. So these are retrograde beads, so they're taken up by the presynaptic terminals and transported back to the original cell body. So when I see these fluorescent cells in the cortex, I know these cells project to the central amygdala, and those are the cells I'm recording from. And I've specifically decided to record in infralimbic layer 5 because that's where I was seeing these projections, these pyramidal projections. And in these studies, what I was actually doing was I was actually looking at cell firing. So I was looking at the circuit activity, and each of these ticks represents an action potential. And what I found was that in alcohol-naive mice versus alcohol-dependent mice, there's actually an increase in the excitability of this projection. So similar to what we saw in the PSTN study, naive mice have projections from PSTN to CEA and medial prefrontal cortex to CEA. And in both cases, alcohol dependence is actually increasing the strength of these connections and the excitability of these connections. And basically, these could be leading to that central amygdala overactivation. And you can see that quantified here, the firing frequency.
Can I ask a question really
Yes.
quick? So, did you look at intrinsic excitability of the cells in the central amygdala, or you just looked at the excitability of the cells from the PSTN to the central amygdala? But these are GABAergic cells that are sending outputs to the PSC, or?
Oh, no. So so to answer your first part, yes. So what we did was, in the previous study, we looked at the PSTN cells that were projecting to the central amygdala.
Right.
And in this study, we're actually looking at the medial prefrontal cells that are projecting to the central amygdala.
Okay.
So this is another input into the central amygdala. And we're looking at the excitability of the projection neurons. And so, so already this circuit is already more excitable in dependent animals. And what was really interesting is when I bath applied norepinephrine. So I said, okay, what do exogenous levels of norepinephrine do to this system? In naive animals, norepinephrine actually decreases the circuit excitability, while in dependent animals, it drastically increases it. So not only is this circuit already more excitable in a dependent animal, or already strengthened in a dependent animal, but norepinephrine greatly increases that activation. And so my hypothesis is this input and norepinephrine's effects in the medial prefrontal cortex may be a significant contributor to that overactivation in the central amygdala. And that overactivation is considered a hallmark of the transition to dependence. So this might be a way to dampen that connection and maybe help alleviate some of those withdrawal-associated symptoms.
The locus coeruleus is what's giving the projection
Mhm.
of norepinephrine. Is it known? Has anyone looked at just electrophysiological activity in the locus coeruleus in alcohol-dependent animals?
Yeah, so that's actually a really interesting point. I was going to bring it up later, but I'm happy to talk about it now. So, so the locus coeruleus is very sensitive to alcohol. And it's a really interesting system because there's also a sexual dimorphism in it. And that links to this study in terms of unique populations. So in naive males and naive females, acute alcohol actually activates the locus coeruleus, as do a lot of other stressors. However, in alcohol-dependent males, this activation is lost. So essentially, locus coeruleus is achieving a tolerance to the acute effects of alcohol. So it no longer responds to acute alcohol. It's no longer releasing norepinephrine across the brain. Females don't have that same tolerance effect, essentially. And so, so that's a really unique sexual dimorphism directly related to alcohol that was another driving factor in this study and another reason I've been pursuing it, especially when you think about these human clinical trials that are using some of the noradrenergic receptor antagonists. It really indicates that we should be thinking about unique populations, male versus females, and comorbidities with stress, things like that. So what we're seeing here is a really interesting effect in terms of the effect on the circuit itself, as well as critically the effect of this neuromodulatory system. And that's, I think, a really exciting thing because it gives us an in terms of how we could think about treating. And as I was just talking about, there are FDA-approved drugs that are used in this system. So the alpha-1 receptor prazosin is FDA-approved. The beta receptor propranolol is FDA-approved. Neither of these for alcohol treatment, but maybe we could think about repurposing. And that's something I'm particularly interested in.
So,
Mm-hmm.
those CEA neurons, they project back to the infralimbic layer, right? Is it layer superficial and deep layer that it projects back to? Right.
So the central amygdala doesn't directly have a lot of connection to the infralimbic.
Okay.
I think it's more prelimbic. But I mean, certainly there are loops in all these circuits. So, the basolateral amygdala receives input from the medial prefrontal cortex, projects, so there's a loop between the medial prefrontal cortex and basolateral amygdala, and the basolateral amygdala also leads into the central amygdala. So there are these big circuit loops.
Yeah.
And that's definitely a complicating factor. But I think I think that's true of a lot of parts of the brain, right?
But is it possible actually to get a slice with both the amygdala and, like you can get like a B1 LGN slice?
Mm-hmm. Yeah, so it is actually possible to get that slice. It's a little bit tricky because you have to cut at a very specific angle, so it's hard to get that slice consistently, but it is possible. But I think this is where the power of, where DREADS and Opto can really help us because we can stimulate specific inputs. And that's sort of, moving forward, that's something that I've done a little bit of before in the past with, the PSTN as an example. But I think it's definitely the power of the technique, and it's opened up a lot of tools for behavior and also for electrophysiology. So what I've shown you so far is that in the dependent animals, there's an excitation of this circuit between the medial prefrontal cortex and the central amygdala, and norepinephrine seems to actually be regulating the circuit, further activating the central amygdala. And the next step in terms of this project electrophysiologically is to start teasing apart the different receptors, subtypes, and which receptor subtypes are involved. But I told you my question is broader than that, and I want to start asking questions about, is this circuit involved in the pathophysiology of the disease, and how is it involved, which is the second part of this project. And so what I've been doing in these same mice is looking at alcohol-induced cognitive deficits and anxiety-like behavior. And so, and this has been my training for my K99, so moving into the behavior and bringing that into my skill set and complementary techniques. And so I'm not sure if you're familiar with the Barnes maze, but it's a fairly commonly used task that is used to assess cognitive impairment. And it's thought to be a task that really recruits the medial prefrontal cortex, which is why I wanted to use it for this study. And so the basic task is that you have a white platform with 20 holes drilled into the sides. And the holes are fairly big, a mouse can climb into them. And the platform is raised. So there's a direct drop between the platform, the hole in the platform, and the floor. And for one of those holes, you actually put a little escape box underneath. So the mouse can climb into this escape box and be out of the bright white light and the fans and the noise that it finds quite aversive. And what we do is we have spatial elements posted around the room so that the animal can orient where it is in the room using these spatial cues and can learn where its box is, because its box is always in the same hole for each animal. And what we can do over time is train the animals to learn where the box is. So they find it very quickly and they make very few errors. And so what we did in this study is first we train the animals where the box is, and you can see the learning curves. And what I've done here is I've separated out the learning curves based on how we then separated the animals. So you can see that all the animals prior to any alcohol exposure learnt the task the same. So this is prior to any alcohol exposure. There are no significant differences in terms of how they learnt the task. So how long it took them to find their escape hole over time and how many errors they made before they found their escape hole. And in this study, we actually had 3 groups because we had animals that were completely alcohol naive, animals that were trained to drink alcohol, and animals that were trained to drink alcohol but also had vapor exposure, so our dependent group. And what we found is that after all the alcohol treatment, so first they were trained in the procedure, then they had their first alcohol exposure and they drank alcohol, or they drank alcohol and were vapor exposed. And then we brought them back to the chambers. And what we found is that on their first day back on the Barnes maze, after all this alcohol exposure, the dependent mice actually had deficits. So they had a higher latency to escape and a higher number of errors that they committed while they were trying to escape. And I just want to show you some videos. At the top, we have an animal that received no alcohol exposure, and the escape hole is over here next to this blue arrow. So he runs in the right direction. The hole is here. And makes one error and goes straight in. And then this is an animal that received alcohol exposure. And for this animal, the escape hole was always here on the right side, so next to this arrow. Oh, yeah, okay. So the hole is here. And you can see this animal knows the task. He knows that he's supposed to be finding the escape hole. And if he can find it, he can escape the bright lights and the white noise and the fans. But he's struggling because the hole is over here. And prior to vapor exposure, he was performing just as well as that air control. So this is a deficit in his memory, in his spatial memory, and his retention of the task. He's still trying. It's over here. He's close now.
What's his current blood alcohol level?
So he's been in withdrawal for 3 days. So the reason we test at a later time point is to make sure that there aren't just deficits because he's drunk. So he's not drunk. He has not had alcohol in 3 days. He's in withdrawal. So this is a good example of cognitive dysfunction in these animals. And it was actually a really exciting finding because it's the first time that anyone has shown cognitive deficits that are related to aversion. So usually when people try to look for cognitive deficits in these models, but when people try to look for cognitive deficits in these models, they look at reward-based tasks, and so then you have to food deprive the animals. So this was really exciting. And so we also looked at 2 other measures. So we looked at changes in digging behavior, and we looked at changes in the latency to feed in a novel arena versus their home cage. And you can see in both cases there were deficits that suggest anxiety-like behavior. And what I haven't shown you is how these tasks relate to this circuit. And that's actually the next step of the project. So changing the activity of the circuit and seeing how that affects the behavior, and also looking at the pharmacological effects. So just really quickly, I'm sorry, I realize we're running a little bit late. Just to wrap up very quickly, what I wanted to show you is that alcohol has these profound effects on stress signaling. And these effects on stress signaling are having effects on behavior, and we can control the behavior by controlling the signaling. And just very quickly, in my future directions, I'm interested in looking at how alcohol and stress affect specific circuits and synapses, how these different peptides, transmitters, neuroimmune elements can impact this circuit activity, and how can we use these different tools to affect the behavior. The 3 main projects I'm interested in pursuing, short-term and long-term, are following up on this K99 project I've told you about, as well as looking at the interleukin-1 beta system and its neuroimmune signaling in alcohol and stress-related changes. And finally, the synergistic effects of alcohol and heat stress in the cortex. And this is work coming out of my graduate work. And all of this relates to cognitive dysfunction. And so I think there's a lot of potential for collaboration with people who are interested in things like aging and mood disorders and sleep disturbance and traumatic brain injury. So thank you for your time. I don't know if there's any other time for any extra questions. Yep.
I'm just interested in what you have in mind for a sleep disturbance.
So that's sort of a long-term goal. It's not something I've had direct experience in. But as I mentioned at the beginning, alcoholics have profound sleep disturbance issues. 8 to 10 years abstinence, they're still reporting it.
So I was
And so I think it's an
jumping
exciting
here,
area.
because I know you get to meet with her in about an hour.
Okay,
Is there
sorry.
anyone who does not still get to meet with her that has any burning questions?
Yeah, go ahead.
Have there been any studies about animals that have more stress related
Have more what, sorry?
Are there any animal studies with looking at like the stress related versus stress related phenotypes and how their alcoholism?
There haven't been studies about that exactly. I mean, I know that's something that's very exciting and interesting in the stress field right now, but certainly stress does affect the alcohol consumption and the behaviors, and so I think that's something that definitely needs to be done.
Thank
Thanks.
you very much for the talk.
Thanks.
We'll see you later.
Sorry, I ran late.
Oh no, they asked like a gazillion questions.